Hello everyone. Good evening to all of you. I hope so. My screen is visible to everyone and voice is also audible to all of you. Right? So, if you remember, in previous week, I was taking the session on Unit-6, that is Plant Physiology. So, certain questions we had discussed, and I had also mentioned that we will take up rest of the questions in my next session. So today we are going to take up the rest of the questions from Unit 6. So are you all ready to take up the questions? Yes? Are you all ready to take up the questions from Unit 6, Plant Physiology? Can I get some confirmation in the chat box? Yes? Okay, fine. So let's take up the first question today for today's session. Following are some statements regarding the synthesis of secondary metabolites in plants. Terpenes are synthesized by shikimic acid pathway and mevalonic acid pathway. Alkaloids are nitrogen containing compounds synthesized by shikimic acid pathway. Phenolic compounds are synthesized by shikimic acid and malonic acid pathway. Both alkaloids and terpenes are synthesized by mevalonic acid pathway and MEP pathway. Here you have to identify correct combination of statements. Thank you, Sarita and Maria, for confirming. So, as I was discussing in my last class, this is also one of the important topic from CSIR NET point of view, that is secondary metabolites. Every time you get questions, either in the form of statements like this, which is there in front of you, or you can expect in the form of match the following. And you'll find that majority of the times the questions comes under part C rather than part B. So, what should be the answer for this question? And recently also you had question from this in the form of match the following. So Unit 6 is actually a kind of unit where very less units or less subunits are ignored. As I was discussing in my last session, it's not that you should ignore nitrogen metabolism and stress physiology, but yes, they are considered to be least important than in other subunits. They are being asked on rotational basis. Yes, I got an answer from Sarita. What about others? Any other answers from anyone else? Try on. This is simply the pathway that leads to the production of secondary metabolites. We have only 3 secondary metabolites. Like categories. I'm not saying only 3. Categories I can say. Alkaloids, terpenes and phenolic compounds. So identify which one is the correct combination over here. Okay, got an answer from Maria and Sarita. Can I expect some more answers? Can I expect some more answers from everyone? Try to participate, everyone. So we are discussing with each other what are the correct and what are incorrect one. So there is nothing wrong in saying incorrect answer as well. It's a learning stage, right? So no problem. Yes. Any other inputs from anyone else? Okay. So the correct answer over here is B and C. That is, alkaloids are nitrogen containing compound being synthesized by shikimic acid pathway. Phenolic compounds are synthesized by shikimic acid pathway and malonic acid pathway. That means statements A and statement D is incorrect. Let's find out how. So this is the overall pathways that leads to the production of secondary metabolites. So you can see primary carbon metabolism produces many intermediates, right? We have erythrose 4-phosphate, we have PEP, phosphoenol pyruvate, then we have pyruvate and then 3-phosphoglycerate. So it is through this intermediates we develop different pathways that leads to the production of different secondary metabolites. For example, for terpenes, you can see that we have mevalonic acid pathway and MEP pathway. MEP is methyl erythritol phosphate pathway. So this can be obtained either from 3-phosphoglycerate or from pyruvate, or pyruvate acetyl coenzyme A gives rise to mevalonic acid pathway. So this is terpenes. If you look into the phenolic compounds, you have shikimic acid pathway and malonic acid pathway, the contributors. Again, malonic acid pathway, get it from acetyl coenzyme A. Shikimic acid pathway, you get it from phosphoenol pyruvate. So they will be producing aromatic amino acids as well and contributor of phenolic compounds. Then the third category will be the nitrogen containing secondary products. That is nothing but your alkaloids. So you can get it from aliphatic amino acids and aromatic amino acids. Aliphatic amino acid, you get it from TCA cycle, and aromatic amino acids, you get it from shikimic acid pathway. So these two are the contributors of your nitrogen containing products. Okay. So these pathways are clear to all of you, or is there any confusion? Based on pathways, you get questions every time. So be clear with the terms specifically like malonic acid pathway, mevalonic acid pathway, methyl erythritol phosphate pathway. Although the name quite similar to each other, but remember they are different. And when it comes to malonic acid pathway, another peculiarity is that this takes place in fungi, whereas shikimic acid pathway takes place in plants. Most of the portions is contributed by your shikimic acid pathway in plants, very less from malonic acid pathway. I hope so I'm clear to all of you. The question and the answer is clear to all of you. Yes, Sarita, Maria. So moving ahead with our next question. Following are some statements regarding the synthesis of jasmonic acid in plants. 12-oxo-phytodienoic acid is produced in chloroplast and transported to peroxisome. Action of lipoxygenase, allen oxide synthase, allen oxide cyclase, it takes place in peroxisome. 12-oxo-phytodienoic acid is first reduced and then converted to jasmonic acid by beta oxidation. Final product of jasmonic acid takes place in chloroplast. Action of allen oxide synthase and cyclase takes place in chloroplast. Now, among the given statements, identify the correct combination. Correct combination of cells. So, jasmonic acid is also one such topic which is considered to be important from examination point of view. So, in my previous class, I had given you some insights regarding other plant hormones like auxin, gibberellins, cytokinin, abscisic acid, and all. Jasmonic acid specifically comes under your defense mechanism. Jasmonic acid and salicylic acid. So you can consider it under secondary metabolites because secondary metabolites are nothing but that helps in the defense responses, right? Okay. Yes, Debashri has given an answer. Okay. What about others? Okay, Maria and Sarita. Anyone else would like to give the answer for this question? So based on jasmonic acid also recently question had come in the exam. So as I told you, one topic that should never be ignored when it comes to CSIR NET exam. Yes, any other inputs from anyone else? What should be the answer for this question? Okay. So let's check out what is the correct answer over here. And you all three are absolutely correct. Debashri, Maria, and Sarita. That is 4. So 4 indicates statement 1, 3, and 5. So how statement 1, 3, and 5 is correct? Let's have a look onto that. First thing is that, can anyone tell me who is the precursor of jasmonic acid? Who is the precursor of jasmonic acid? Because you have precursors for every plant hormone, right? Like if you take auxin and ethylene, they are amino acid derived hormones. Then when it comes to rest of the hormones like cytokinins, trigolactin, gibberellic acid, for them they have terpenes. So when I say jasmonic acid, who will be the precursor? Absolutely correct, Sarita. This is linolenic acid. Yeah. So this linolenic acid will give rise to, it undergoes several intermediate steps, giving rise to 12 oxo-phytodienoic acid, which in turn gives rise to jasmonic acid. So the only plant hormone to have a lipid serving as a precursor. So these are the steps involved in the production. So you can see we have 2 cell organelles involved in the jasmonic acid synthesis pathway, that is chloroplast and peroxisome. So in chloroplast, you can see that linolenic acid in the presence of lipooxygenase, allen oxide synthase and allen oxide cyclase gives rise to an intermediate product known as 12-oxo-phytodienoic acid. So this product is synthesized where? Within the chloroplast itself. And from here, this product is released into the peroxisome, which undergoes reduction reaction and beta oxidation to give rise to jasmonic acid. So this is how the synthesis goes for jasmonic acid. Now, if you look into the signal transduction pathway, for that an amino acid is there. We have isoleucine combined with jasmonic acid. So in the presence of a protein known as JAR1, these two proteins, that not proteins, jasmonic acid and isoleucine will be combined together as a complex. Coordinating them, we have the SCF CoA1. So what does this SCF will do? SCF is responsible for adding ubiquitination. Where they will be adding ubiquitination? To the corresponding repressors. Because if you remember, in my previous session also I said you, whenever you study a signal transduction pathway, 2 things are important to be remembered. First is the receptor. Where are they located? Then who is the repressor in that pathway? And then who is the transcription factor in the pathway? So these are the 3 major aspects of any signal transduction pathway. So when it comes to jasmonic acid, JAS is considered to be the repressor and MYC2 is considered to be the transcription factor. So if JAS is polyubiquitinated, then MYC will be free. If MYC is free, transcription will be initiated. So that's how it goes. This is the diagrammatic representation of the same. So you can see jasmonic acid and isoleucine combined together as a complex with SCF CoA1, right? So now this forms an entire complex. Now this complex, what it will do? It will go and bind to the repressor. So JAS is the repressor, MYC2 is the transcription factor. So this entire complex will go and bind to JAS removing it from the transcription factor MYC2. And the JAS will be tagged with ubiquitin tags so that they will be targeted for degradation. Now, since the JAS is removed, your transcription factor is free, so they are ready to carry out the JA dependent genes, that is Jasmonic acid dependent genes, including those that are involved with defense mechanism as well. Okay, so based upon signaling, they can ask you question. Based upon the synthesis part also, they can ask you the question. So, so remember, be clear with this protein's name, not to be confused. JAR1 is just an accelerator to form this combination, that is combining the jasmonic acid with isoleucine. JAS is the repressor here and MYC2 is the transcription factor here. Okay. So whenever you're studying signal transduction pathway, it is good that if you understand through images, that will be more easy for you. Next, moving on to our next question. Dark grown Arabidopsis seedlings, when exposed to ethylene gas, shows typical triple response. So here are some statements regarding the triple response. A dominant ethylene receptor mutant will not show a triple response in the presence of ethylene. Tightening of apical hook is one of the feature of triple response. Loss of function of multiple receptors will show triple response even in the absence of ethylene. Increase in the hypocotyle length is a feature of triple response. Now identify the correct combination of statements from here. So ethylene based question. So in our last class, I think we had discussed about abscisic acid, right? In our last session. So taking into consideration individual hormones, they can ask you a question, or you can expect questions in a combined format. Combined format means miscellaneous hormones, functions they might be giving you, and then you have to identify the correct answer from there. Yes, okay. Debashri has come up with one answer. Okay, what about others? So who is the precursor of ethylene? Who is the precursor of ethylene? So you all might be aware that there we have only two plant hormones which is having amino acid derivative, right? So auxin, we have tryptophan. What about ethylene? Yes. No. Indole acetic acid, IAA is your auxin, no? Tryptophan will be serving because they have their structure similar. Indole ring will be there in the structure. Then, yes, you are correct, Maria. We have methionine as the amino acid serving as the precursor for ethylene. Correct. Yeah. No issues. At least you will not make the same mistake again, no? So, yeah, what should be the correct answer over here? So, I got answer from Debashri and Maria. What about Sarita? Any answer from your side? And what about others? Try to participate, everyone. Try to interact, everyone. Okay, I got 3, 4, 3. 3 from Debashri, 4 from Maria, and 3 from Sarita. Anyone else? Anyone else would like to give the answer here? You have to be very clear with what is a triple response with respect to plant hormone. Secondly, you have to be clear with the mutation of ethylene receptor. So in your Tays and Ziegler book, you have this diagrams represented very nicely over there. Okay, I got 3, 4 and 1. So let's find out what is the correct answer over here. Yes, you are absolutely correct. The correct answer for this question will be statement A, B, and C. D is incorrect because increase in hypocotyle length is not a feature of triple response. Rather, it should be decrease in the hypocotyle length. So rest all the statements are correct. C. Triple responses generated by etiolated seedlings include reduced stem elongation, increased lateral growth, that is the swelling part, and there will be abnormal horizontal growth of epicotyle. So these are the three features of your etiolated seedling triple response. Now, if you look into the model for ethylene receptor based upon their receptor mutant, there are four models mentioned over here. So, what is the unique feature of ethylene signaling pathway is that majority of cases you will find that when a ligand goes and binds to the receptor will be activated and they will generate a response. But in this case, what happens? You have to remember the other way around, because when the ethylene goes and binds to the receptor, and where is the receptor present? See, in the ER. So when the ethylene goes and binds to their receptor, the receptor is inactive. That is shown in the form of a lock position. And because of that, we are getting the ethylene response. So that is a unique feature for ethylene binding. Now, if you look into the second model, all the receptors you can see, there is no ligand present, right? Because of which your receptors are active now. Now, since your receptors are active, they are not able to generate any response, that is suppressing the ethylene response. Now, if you look into the third model, so we have 5 receptors for that. Suppose there occurs a missense mutation to one of the receptor. Missense mutation means a change in the amino acid. So what happens is that the response does not occur and the single constitutively active receptor will be sufficient to shut down the ethylene response. See, if a single receptor is mutated, that means they are activated, then also you won't be able to get a response even though the other receptors are inactive over here. Okay, and the fourth one is the disruptions in the regulatory domains of multiple ethylene receptors. So that means at least 3 receptors are mutated. So in that case, what happens? Constitutive ethylene response will be generated. Okay, so these are the 4 different models for your ethylene receptor action. I hope so I am clear to all of you. No confusion, right? Yes, the triple response and the 4 models of ethylene receptor. Clear, right? Can I get a thumbs up? Yes? Okay. So this is just a reminder that we have a Lakshya batch going on. So for the upcoming CSIR NET exam, that is CSIR NET 2025. And in this batch, we have unlimited classes. We have 24/7 chat support system. We have study portal. And also you'll be having access to molecular biology free internship program as well. Okay. Then we are also providing with some smart tools like Conceptica series where you'll be able to access the comparison tables, the flow charts, the mind maps and all. Then we will have regular test series going on, that is weekly test, monthly quest. So that will help you to make yourself better in terms of preparation. So if you have any query, you can dial to our toll-free number or yes, Maria, online classes are available. Or you can visit at stores.biotechnica.org. We have both morning and evening batch available. So whichever you feel you can check with the check in through this toll-free number. Okay. Moving on to our next question. So again, this is related to secondary metabolites. So you can see Some plant natural products are given on as list 1 and their basic structural unit is provided in the list 2. So identify the correct combination from the given options. See, it is not simply the secondary metabolite function aspect is important, but at the same time, their structural feature is also equally important. And this was a recent question in the CSIR NET exam. What should be the correct combination over here? Phenolic compounds, alkaloids, terpenoids, cyanogenic glycoside. On the other hand, you have 5 carbon isoprene unit, glucose unit attached by O beta D glucosyl linkage. Then you have nitrogen containing compound, then aromatic arene ring with OH group. What should be the answer over here? Okay, I got an answer from Sarita and Maria. What about others? Anyone else who can make up this answer? Unit 6, one another feature is that you always expect direct question. Majority of times. So if your concepts are clear, so unlike other units, if you take where experimental questions are being asked on a regular basis, Unit 6 is something which gets questioned specifically from one book, that is Stays and Zigger. If you are clear with your concept, if you are clear with this reference book, then no one can stop you from getting highest marks in plant physiology part. Very rarely you get some kind of experimental question. Okay. Yes, Debashri also gave the answer. Anyone else? Anyone else? You all are absolutely correct. So 4 is the answer. So phenolic compound is then having an aromatic ring with OH group. Alkaloids is a nitrogen containing compound, which we have discussed in the first question. Then terpenoids, these are having 5 isoprene units. So isoprene serves as the precursor for many plant hormones, which also I had mentioned. And lastly, it is your cyanogenic glycoside, which is having a glucose unit attached by O beta D glucosyl linkage. So in this combinations, if you see, if you are confident that phenolics is having the aromatic ring, you can easily eliminate option 2 and option 3. Now you have to just find out among these two which is the correct one. So majority of the match the following questions, you can work out by eliminating the options. Sometimes that unique combination will be there as one such option. So you have to have a look onto that as well. Moving on to our next question. Young seedlings of Arabidopsis plant is exposed to the following light conditions. Far red light followed by red light, far red light followed by red light, then a dark phase, red light followed by far red light, dark phase followed by far red light and then red light, far red light followed by dark phase and then a red light followed by dark phase and then a far red light. So you have to identify the correct conditions that leads to photomorphogenesis. I hope so you all are aware about photomorphogenesis. Are you all aware of scotomorphogenesis and photomorphogenesis? Are you all aware about scotomorphogenesis and photomorphogenesis? What is the difference between them? What is the major difference between them, in fact? Yes, scoto indicates night and photo indicates in the presence of light. So, what should be the correct answer over here? So, this itself, this experiment itself is given in your Tays and Ziegler book. Yeah, Sarita, I saw your answer. Then, what about others? Can you tell me, Sarita, why did you opted for option 4? Option 4 is what? Statement A, D, and E. Is there any specific reason why did you opted for only these three statements? Why not any other statement? Meanwhile, let others give me the answer. Yes, what should be the answer for this question? Maria, Debashri, others, whoever is watching this session. Okay, red light causes PFR to PR conversion. Confusion between 1 and 4. Okay. PR to PFR. Yeah, PR to PFR. So, which one is the activated version, PR or PFR form? I think we had discussed in our last class with respect to gibberellins. Which one is the active version of this phytochrome? PR or PFR? With respect to gibberellin, PFR is the active version. Yes. Any answer from your side, Maria? So Debashri said she has a confusion between 1 and 4. And Sarita has given 4 as the answer. So, Maria, do you have any answer from your side? So, the correct answer over here is what you said is absolutely correct. It is statement A, D, and E. First thing is that, no problem, Maria. Now you will be clear with this question and you will understand how to get into a conclusion. So, remember, photomorphogenesis means what? When a seedling develops in the presence of light. So, apical hook will open, or we can say coleoptile splits open. So, in the light, obviously, leaf growth will be promoted, chlorophyll development takes place, stem elongation takes place in the night. So, here it will be suppressed. Radial expansion of stem takes place, root elongation takes place, and lateral root development takes place. So, these are the features of de-etiolated plant, or I can say plant response, light responding plants. Now, contradictory to that, if you see the scotomorphogenesis part, it is when a seedling develops in the dark condition. So, if you are remembering just one, you will be able to remember the other. So, it is not necessary that you should remember the features of both photomorphogenesis and scotomorphogenesis. You can remember one just to avoid confusion, because all the features that are mentioned over here will be opposite. For example, here we have a distinct apical hook. There won't be any leaf growth, no chlorophyll, rapid stem elongation. So there it was suppression. So here elongation. Rest everything will be limited. Limited radial expansion, limited root elongation, limited lateral root formation. So whichever you feel confident, remember only that part. The other part, you can remember it as an opposite one. So now coming into the experiment, why it is A, D, and E are correct. So there was an experiment carried out with the seedlings in the presence of PR and PFR. So what they found that when the seeds were exposed to dark condition, they were not germinating. But when they were provided with the red light, they were germinating. Again, they exposed it to far red light, germination reduced. And again, when they give rise to red light, like red light, far red light, and then red, germination was accelerated. Again, they tried to give the far red light. The germination were inhibited. So, from this experiment, they concluded that red light is the one that carries out seed germination, whereas far red light is the one that inhibited the seed germination. So, with this discovery, we can conclude the statement A, D, and E as the correct. So, remember, if you look into the statements, everything in all the statements, it ends with red light. And I said you that red light is an activator of seed germination. So, that's the reason statement A, D, and E are correct. I hope so I'm clear to you now, Debashri and Maria, without any confusion. This experiment, yes, Debashri and Maria, I am clear to you, both of you. Okay, fine. Next, moving on to the next set of match the following kind of question. Now, this is related to the PR proteins present in plants with their activities listed in column X and column Y. You have to identify the correct combination from here. Are you, do you have any idea about PR proteins? Do you have any idea about PR proteins? Have you heard about this PR proteins? What are the PR proteins, or when are they synthesized in the plants? You have not heard about it. What about others? Have you heard about PR proteins? Okay, Debashri and Maria have not heard about this proteins. What about Sarita? Have you heard about this PR proteins in plants? Okay, Sarita has given answer. So do you have any idea about PR proteins in plants? Anyone else would like to give the answer for this question? Again, this is the part of secondary metabolites. Pathogenesis-related proteins, that's what we call it as PR proteins. That means, as the name indicates, these proteins will be synthesized during some extreme conditions. Right? So, the correct answer over here is okay, fine. No problem, Sarita. So, you told answer correctly. Yes, you are correct. So, we have different types of PR proteins, and based upon the PR proteins, you have got the corresponding features. So, for example, PR2, they are having beta 1,3 glucanase activity. PR3 is chitinase. PR6 is proteinase inhibitor, and PR10 behaves like a ribonucleases. So every protein has some unique activities that are required by the plants for their survival. See, pathogenesis related proteins are a group of proteins produced by plants in response to pathogenic attack, mechanical damage, or even exposure to stress factors like UV light, drought, or if any kind of chemical treatment given. Apart from that, They can also be induced by signaling molecules. That means specific hormones like salicylic acid, jasmonic acid, even ethylene. They play a crucial role in plant defense mechanism, and they are part of plant's innate immune system as well. As I told you, these proteins are classified into 17 families based upon their amino acid sequence, their enzymatic activities, and their biological function. And if you see, each PR protein will have unique feature and they carry out unique role in defense mechanism. So if you ask me, where are these proteins located? Well, they are located in different parts of the plant. They can be located in apoplast, that is the cell wall space. They can be located in vacuoles, cytoplasm, even extracellular space where they can directly interact with the pathogens. Now, looking into the types of PR protein, these are. So, PR1 to PR17. So, different PR protein is having different function. PR1 is antifungal. 2 I have mentioned. 3 also I have mentioned. 3 and 4 both are chitinase but of different types. Then 5 is there, 6 is there till 17. So based upon the PR protein, different properties. So this table is again important from examination point of view. You can expect question like the way I had asked you, that is the direct question tabular form. Sometimes they will provide you with the same question in the form of statements, wherein you have to identify the correct combination of statements. Okay. Next. Yes, this is a question related to stress physiology. So they are provided with some abiotic environmental factors in column X, and what is their after effect in plants. Identify the correct combination here. So as I told you, stress physiology questions are very less or very rare. You get sometimes you will get, sometimes you will not get. This is a recent question. So in column X, we have factors, that is water deficiency, salinity, flooding, and high light intensity. On the other hand, for column Y, we have iron cytotoxicity, hypoxia in the roots, photoinhibition, and then water potential reduction. So what should be the correct combination over here? Okay. Nagashri, Debashri, and Maria. So 4 answers. Any more answers? Anyone else? Good to see more participants interacting to give the answers. See, I always say whether you are giving a right answer, wrong answer, no problem. But the point over here is to give the answer. Anyone else? No? So this time I got 4 answers. So let's find out the correct answer over here. In fact, this is a very easy question. Even if you have not studied stress physiology also, you will be able to get into an answer for this question. So the correct answer, as you all have mentioned, yes, it is 2. So water deficiency results in water potential reduction. Salinity causes iron cytotoxicity. Flooding causes hypoxia in the root system, and obviously high light intensity will cause photoinhibition because this can cause damage to the cell organelles. So water deficiency stress can be defined as a situation where plant water potential and turgor potential will be greatly reduced to interface with normal functions. Salinity will also have the same effects as that of water stress in plant growth, excepting that one extra point over here is addition of iron cytotoxicity, that is because of the presence of salt in the soil, excess of salt in the soil. Then, when the soil becomes waterlogged or flooded, the air spaces in the soil will be filled with water, thereby restricting the diffusion of oxygen to the roots. So, as a result, what happens? Roots will be devoid of oxygen. So, this leads to the condition we call it as root hypoxia or anoxia condition. That means where oxygen is not present. And the last one is photoinhibition. As I told you, it is the process of reduced photosynthetic activity and damage to the photosynthetic machinery due to excessive light exposure. That means, obviously, the cell organelle, that is your chlorophyll, chloroplast, everything will get damaged because of excessive light. If you remember, in my previous session, I have also mentioned you about the role of FORT proteins, FORT1, FORT2, and how they are helping in avoidance response, elimination response, and all. Right. So when the light intensity is low, all the chlorophyll molecule will accumulate at the top to receive maximum light. But when the light intensity is higher, they will accommodate towards the side of the cell so that they should not get damaged. So that was carried out by phototropins. Next, here are some statements regarding the materials transported through the phloem of a plant. Only reducing sugars are translocated. Non-reducing sugars are generally translocated. Sucrose and raffinose are generally translocated. D-glucose and D-fructose are translocated. Identify the correct combination of statements from here. So this is related to solute transport. What should be the correct combination over here? Okay, Sarita and Maria has given the answer. What about others? So again, solute transport, if you compare it with stress physiology and nitrogen metabolism, you get better number of questions from solute transport. Okay. So the priority should be based upon the importance of each subunit. So you have got subunit 6A to 6H, right? So based on that, you have to channelize or idealize the preparation. Okay, yes, Debashri and Lakshmi Prabha has also given the answer. Anyone else? Anyone else would like to give the answer for this question? So sometimes you can also expect questions upon the transport mechanism, apoplast, implast, transcellular pathway, like that. So be clear with the terminologies so that you will be able to answer it. Anyone else would like to give the answer? Okay. So the correct combination over here, as mentioned by majority of you, B and C. So that means non-reducing sugars are generally translocated through the phloem. So we have examples like sucrose and raffinose. C. So basically, we have got three cells, and this model is known as polymer trapping model of phloem loading. So what does it indicates? We have bundle sheath cell, we have intermediary cell, and we have sieve elements. Intermediary cells are specialized companion cells which are located adjacent to the sieve elements in the phloem, connected to the sieve elements by numerous plasmodesmata and also helps in the movement of nutrients as well as some signaling molecules or signals between the cell. So what happens here is we have the sucrose molecule constructed in the bundle sheath cell. So sucrose is constructed by a combination of glucose and fructose. Right? Glucose and fructose combines together giving rise to sucrose. Then from here through plasmodesmata, they will be released into the intermediary cells. Now, into this sucrose, another sugar is also added over here. We call it as galactinol. So, when you are adding galactinol to this molecule, to the sucrose molecule, it becomes a bulky molecule now. So, why they are making it a bulky molecule? So that the back diffusion to be prevented. So, now what you got? Raffinose plus inositol. So, we can ignore the inositol part. Raffinose is important. Now, this raffinose can easily diffuse into the sieve elements. Okay. So, sucrose synthesized in the mesophile diffuses from the bundle sheath cells into the intermediary through the plasmodesmata. Within the intermediary cell, raffinose will be synthesized. How? From sucrose and galactinol. And they will maintain the diffusion gradient for sucrose. So, because of its large size, back diffusion will be prevented. So, they will be moving ahead to the sieve elements. So, apart from raffinose, we have also stachyose that can move along the in this different types of cells. But for convenience, we have just shown only raffinose here. Okay. So, like that, the sugar will be transported from here. Next, what we are having? Next question. So, that is how it goes. So, the next question. Here are the list of plant hormones with their precursor. Identify the correct combination here. Very simple and easy question. Correct combination of plant hormones to that of their precursors. I was also mentioning you about the precursors also, right? They are also important from examination point of view. So you have auxin, cytokinin, ethylene, gibberellin. Geranyl diphosphate, adenosine molecule, tryptophan, 1-amino-cyclopropane, 1-carboxylic acid. Okay. I got answer from Lakshmi, Sarita, Maria, and Debashri, Nagashri also. So miscellaneous answer 2, 3, 1. 2, someone has said 2, someone has said 3, someone has said 1. Anyone else would like to give any other answer or contributing to any of this answer? That's it? Okay. So the answer for this question is, the correct answer over here is 3. So it is neither 2 nor 1. First thing is that, as I told you, we have 2 plant hormones which are derived from amino acid. But here they have very cleverly mentioned as 1-amino-cyclopropane, 1-carboxylic acid. Right? So this is ACC, and ACC is considered to be an immediate precursor of ethylene. It's not the direct precursor. The direct precursor will be methionine. But ACC is considered to be the immediate precursor of ethylene because they carry out or they catalyze several processes, ACC synthase. So auxin, you can say it is tryptophan, right? You can say it blindly. So one option and fourth option you can easily eliminate. So there is no point of saying option 1 and option 4. Now what is remaining? 2 and 3. Then you have got cytokinin. If you remember, cytokinin, most of the examples of cytokinin, that is naturally occurring versions and the synthetic versions of cytokinin, all these are adenosine derivatives. Whether you take into consideration kinesin, zeatin, their examples all are adenine derivatives. So B to 2. Now you are on the safer zone. Then ethylene, as I told you, this is an immediate precursor. Gibberellines, they can be derived from your terpenes. From terpenes, we can get geranyl diphosphate. So you can consider this also as a precursor. Okay. So that is how you can get into the answer for this question. Not difficult, but yes, you have to think a bit to get into a conclusion. Okay. Moving on to our next question. Following are some statements regarding the plant growth hormone ethylene. Climatic fruits exhibit a characteristic respiratory rise before the ripening phase. Non-climatic fruits exhibit a characteristic respiratory rise before the ripening phase. In the leaf shedding phase, higher auxin concentration increases the sensitivity of abscission zone cells to ethylene, whereas in the leaf maintenance phase, higher auxin concentration increases the sensitivity of abscission zone cells to ethylene. Identify the correct combination of statements from here. What should be the correct combination of statement from here? In this question, you have to be clear with two things. What is the difference between climatic fruits and non-climatic fruits? First thing. Secondly, you have to understand the concept of leaf abscission process. What is the shedding phase? What is the maintenance phase? What is the abscission phase and all? Then you will get into a conclusion. Can anyone tell me what are the two plant hormones which participate in the leaf abscission process? The two plant hormones which participate in the leaf abscission process. Which two plant hormones? Yes, you are correct, Maria. It is auxin and ethylene. Now, identify the correct combination over here. Okay, the Bishri has given the answer. What about others? What about others? Ethylene and abscisic acid will not come in the abscission zone. Ethylene and auxin only will come in the abscission zone. Nagashri has also given option 1 as the answer. What about others? Maria also opted for 1. Sarita, do you have an answer for this question? Okay, let's have a look into the correct answer. The correct answer over here is A and D. So let's check it out why these two statements are correct. So the first thing is that you have to be clear with the different phases of leaf abscission. So as I told you, we have auxin and ethylene that plays a very important role in the abscission process. So we have leaf maintenance phase, we have the abscission induction phase, and then we have the abscission phase. So in the leaf maintenance phase, we have higher auxin concentration. So if auxin levels are higher, the leaves will appear green in color where they will be inhibiting the ethylene response. In the abscission induction phase, that is in the initial stage, you will find that there is light yellowish in color. So at that stage, what happens? There is reduction in the auxin level and there will be increase in the ethylene response. And the final phase will be the abscission phase, where it will be complete yellowing of the leaf and shedding off because of the digestion of the cell wall, because they have specific enzymes present that will help in the degradation of their cell wall, and like that the leaves will fall down. So that is after complete yellowing of the leaves. So the leaf death takes place. Okay. So this is how you need to get into an answer. So before winding up the session, I just wanted to remind you, for Unit 6, never ever skip subunit A, subunit B, that is related to photosynthesis and respiration, then the plant hormones part, then the sensory photobiology, then secondary metabolites, and next to that is solute transport, and after that you will have nitrogen metabolism and stress physiology. So remember this order whenever you prepare for the exam. So in the next session, I'll come up with Unit 12. Prepare yourself and let's have another interactive session in the next week. Thank you so much, everyone. See you again in the next session.